Let's take a look at active transport. Active transport needs energy in the form of ATP. The type of molecules that usually use primary active transport are ions that are large, polar, and cannot pass through the phospholipid bilayer. So they need some kind of a protein, a membrane protein. In active transport, molecules are moving up the concentration gradient and therefore they need a protein pump. So there's the protein pump in red. And this pump must be powered by ATP. Let's label the outside and inside of the cell. So let's imagine the outside of the cell has fewer solute molecules when compared to the inside of the cell. So this solute could be sodium, potassium, calcium, magnesium. Any of these ions use this type of transport to move from a region of low concentration to a region of high concentration. So let's say the inside has high concentration and the outside has a low concentration of this particular solute. Because we're talking about active transport, the movement of molecules is going to go from low to high. We call this up the concentration gradient because you're going from low to high. So it's up the concentration gradient, and you know whenever you go uphill, you're going to use more energy, which is why we need ATP. We call this type of transport primary active transport. Another kind of active transport is called secondary active transport. The purpose of secondary active transport is to use the energy derived from the movement of sodium into a cell to carry along with it other molecules such as glucose, amino acids, and other ions. So, secondary active transport allows the movement of glucose, amino acids, and ions into a cell by catching a free ride with sodium. So let's see how that happens. Let's take a close look at secondary active transport. Secondary active transport takes place in many cells in the body. For our purposes, we'll take the example of the cells lining the small intestine. If you remember, the cells lining the small intestine are made up of simple columnar epithelial cells. They are long cells, which means they have a front end and a back end of the cell. And these two ends are quite different. So there's the front end. It appears to have a channel, and the back end has a protein pump. So there we have the channel in the front end, a protein channel, and the back end has a pump that is powered by ATP. This secondary active transport depends upon the gradient of sodium. There's always a lot more sodium outside the cell than inside. This allows the sodium to rush into the cell in a kind of whoosh. It's a huge amount of energy that is derived from this movement of sodium. This energy is used by other molecules to catch a free ride with sodium. For example, we have glucose. Glucose enters cells in this way by using sodium, the movement of sodium, to enter. Now, if this keeps continuing, sodium keeps entering, the concentration gradient of sodium is going to start decreasing because the sodium inside the cell is going to go up. And the difference between the outside and inside is going to decrease. So, how do we solve that? This is solved by pumping the sodium out via the back end of the cell because there is a protein pump. That pump moves sodium from a region of low concentration to a region of high concentration, which is outside. So the primary active transport that you see at the back end is what makes the secondary active transport at the front end possible. Let's take a look at bulk transport, which is the third kind of active transport. It requires ATP as well. Bulk transport, as the name indicates, is the movement of parcels into and out of the cell. If the package is entering the cell, then we call it endocytosis. If the package is leaving the cell, then we call it exocytosis. Endocytosis has two types. If the cell is accepting a package with solid particles, then we call it phagocytosis, or the cell eating. If the cell is accepting a package with liquid particles, then we say that it is pinocytosis, or the cell drinking. All of these types of bulk transport require the use of ATP for energy. Let us take a closer look at endocytosis and exocytosis. The structure you see in yellow is a virus. This is a white blood cell in pink. It's a macrophage that recognizes the virus and wants to take it in and kill it. The membrane of the macrophage has these little red structures which are receptors that recognize the virus. The cell then engulfs the virus and puts it into its lysosome, which then destroys it. We call this process endocytosis. Most specifically, we call it receptor-mediated endocytosis because the receptor helps to recognize the virus. And the type of membrane protein that is important here is a receptor protein. The next process is exocytosis. Let's imagine that this cell is making antibodies, which are proteins, against that virus. So it puts the antibodies in a package called a vesicle and brings the vesicle to the cell membrane. At the cell membrane, there is a linking protein shown in red, and that helps to link the vesicle to the cell membrane and expel its contents. So in exocytosis, the membrane protein involved is a linking protein.